Okay, afternoon everyone. So I'm here to introduce Professor Verhaak for the Brain Cancer Seminar Series. So I'm sure most of you in the audience know about Professor Verhaak, but I'll just give you some background. So he undertook his postdoctoral training at the Broad Institute and the Dana-Farber Cancer Center before establishing his research group in MD Anderson in 2010. After that, he was affiliated with the Jackson Laboratory for Genomic Medicine and then moved his laboratory to the Yale School of Medicine in 2023, where he now resides as the Harvey and Kate Cushing Professor in the Department of Neurosurgery. He's a recipient of many prestigious awards, including the AAAS Watchell Award, the Agilent Early Career Professor Award, and the Peter Steck Memorial Award. His lab now studies tumour evolution and mechanisms of therapy resistance in low-grade gliomas, and the group was foundational in establishing the GLASS Consortium, so that's the Glioma Longitudinal Analysis Consortium, which has established a valuable resource of molecular profiles over time in a large cohort of glioma patients. I think it's a fantastic example of decentralized shared science, and the resource has been vital in helping identify and describe genetic scars and cellular phenotypes associated with glioma progression and disease recurrence. His laboratory now focuses in part on extrachromosomal DNA amplifications and their role as critical drivers in tumor progression. He's the co-founder of Boundless Bio, a company that develops ecDNA-directed therapies for oncogene amplified cancers. So I'm very excited to hear more about this work and welcome him here today for his talk around the molecular base of therapy resistance in glioma. Thank you very much.
So thank you for that very generous and kind introduction. It's a pleasure to be here. I just actually came from the US this morning, and I'm originally from the Netherlands, in case you couldn't guess by the t-shirt. I hope everybody's as excited to watch today's games as I am. But I realized how Americanized I've become because I asked the staff in the cafe if there's any place that has AC, and they laughed so hard at me, and I totally deserve it. I changed up the title of my talk a little bit because I'm very excited about this particular set of data. It's, I think, a nice rounded story. We're going to hopefully submit the associated manuscript pretty soon. It's actually not that long, so I think we're going to maybe have a little bit of a shorter seminar. I don't know if you usually are used to an hour, but this is probably 30 minutes. And then everybody can go off to lunch. I'm going to talk specifically about IDH mutant low-grade glioma. In part because I think this is actually a type of glioma that may not get the attention it really should, it deserves. Even though with the recent Indigo trial and the upcoming approval of a new therapy, there's more attention for it. And I think it deserves more attention in particular because it's a tumor type that affects younger individuals who will often succumb to disease earlier in life than their counterparts with an IDH wild-type tumor who get diagnosed later in life. So just to make sure we're all on the same page, I'm talking about IDH mutant low-grade gliomas. These are relatively slow-growing but still very deadly tumors. After an IDH mutation is detected in a glioma, these tumors get further separated into those that carry a 1p92q co-deletion, and these are a majority oligohistology tumors. And then there are their counterparts that do not contain the 1p92q co-deletion, and these in majority contain the astrohistology. I'm going to refer to those as IDH-O and IDH-A in the rest of my talk, probably. You'll see those labels on the slides. So, patients with an oligodendroglioma their median age at diagnosis is 45 years. And they have a great survival relatively, 17 years. But if you add up these numbers, you'll see that that still means that patients die well before retirement. And this is even more pronounced for patients who have an IDH mutant non-codeleted glioma, where the median age of diagnosis is just 37 and their survival is a poor 5 years. So even though we generally consider IDH mutant gliomas to be almost more favorable disease, it really isn't. It really is more, it has actually a greater impact on patients and their families than patients who have an IDH wild type glioblastoma, even though of course it's a horrible comparison to make. So we're talking about two different types of glioma, two different types of IDH mutant glioma, those with 1p19q co-deletion and those lacking a 1p19q co-deletion. This is a paper that's now almost a decade old, published by TCGA. I think most people still know what TCGA is. This is actually a fairly fundamental paper published back-to-back with a paper from Bob Jenkins' group from Mayo, where TCGA, we and others, did genomic profiling across IDH mutant glioma and showed that these tumors contain vastly different sets of molecular alterations, are driven by vastly different genomic abnormalities. For example, IDH-Os are almost universally contain a mutation in the TERT promoter, which is lacking in the astrocytomas, whereas the astrocytomas are universally driven by p53 mutations, which are lacking in the oligodendrogliomas. So the fundamental change in these tumors is the IDH mutation, but after that after that founding event has taken place, these tumors almost go into a different trajectory in terms of further pathogenesis. At least that's what these different sets of molecular abnormalities seem to suggest. Now, our colleague Mario Suva and Hitai Tarosh and many others have done fundamental work in single-cell analysis, including single-cell analysis of IDH mutant tumors. And when they published their sets of papers a couple of years ago, they highlighted that you could project single cells derived from an IDH mutant glioma on an inverted y-axis. And this inverted y is showing on one axis stemness scores with higher scores being more stem-like. And on the x-axis, a axis that goes from astrocytoma differentiation to oligodendrocyte differentiation. And they showed in their work that when you do single-cell analysis combined of codels and non-codels, that actually all of them contain cells that are both stem-like, astrocyte-like, and oligodendrocyte-like. So at the single-cell level, and by looking at single-cell signatures, there's actually much less difference between these two types of glioma than you might have anticipated based on their vastly different sets of genomic abnormalities. Another point to make when looking at this plot is that as far as there are cycling cells in these tumors, they are all found in the group of cells that have a stem-like signature. So the three major cell states that Mario and Itai have described are neuroprecursor cells, which are here at the top, and as I said, the astrocyte and the oligodendrocyte-like, which are in majority non-cycling cell types. So that's sort of the basis of IDH mutant glioma in terms of molecular features, in a nutshell. Now, by the time that we diagnose these tumors, we're looking at a tumor that has undergone potentially decades of evolution, starting from one single cell of origin, but then after that cell starts to proliferate more quickly than the cells surrounding it, some of those cells will be able to better respond to challenges in the microenvironment, for example, hypoxia. And because of that, by the time of diagnosis, we're looking at a tumor that has a high amount of intratumoral heterogeneity. And of course, this intratumoral heterogeneity doesn't end at diagnosis. In fact, I would argue that because of the treatments that we give to the patients with these tumors, actually we're enhancing that evolutionary process because we're putting new stresses on the tumor, not just by debulking the tumor, but also by irradiating it and by providing by giving these patients chemotherapy. So the three major treatments for these patients are, of course, the bulking surgery, where safe resection is possible. Then radiation, which has great benefits. It's actually a very effective therapy for these patients. But of course, there's many downsides, including the cognitive deficits that can be derived from radiation. So more comorbidity. And then there's chemotherapy, which is often temozolomide, which really is quite ineffective, but also has relatively low amounts of comorbidity. So therefore, it's often selected as a treatment. And each of these treatment arms have their own evolutionary bottleneck effect. We and others have shown that if you evaluate mutational burden of patients before they receive temozolomide and after they receive temozolomide, then in a subset of those patients, the tumor has become hypermutated under the influence of the temozolomide. So if you look at this plot, this is from a 2019 paper from our lab in collaboration with the GLASS Consortium. These are pre and post mutational burdens on a log scale. So you can see we drew the line at about 10 mutations per megabase. And you can see that a subset of patients in both the CODEL group and the non-CODEL group, their tumors have become hypermutated. And this is an effect of temozolomide, and this is an example of how tumors continue to evolve after diagnosis. Similarly, a couple of years later, we evaluated different types of mutations before and after radiation, and found that under the influence of radiation, IDH mutant gliomas acquire small deletions, microdeletions that range in 2 to 20 base pairs in size, scattered across their genome. So radiation doesn't drive point mutations, radiation drives the formation of microdeletions. Another type of genomic abnormality that is frequently acquired after tumors have been diagnosed is the homozygous deletion of CDKN2A, and in fact this has now become a pathology marker for a tumor that is classified as a high-grade disease. And it's really most relevant for non-CODEL tumors. So these are CDKN2A hemizygous and homozygous loss statistics at diagnosis in oligodendrogliomas and at recurrence in oligodendrogliomas. You see there's a small increase, but not very significant. Whereas for the astrocytomas, there's well over a doubling of the number of cases that have either a hemizygous or a homozygous loss of CDKN2A. Over half. Now, what's important here is that if you then evaluate how often CDKN2A is acquired as a deletion in the context of radiation, we see that actually after radiation, over 2/3 of tumors have acquired a hemizygous or homozygous deletion of CDKN2A. And this frequency is less than half in patients who have not received radiation. So radiation isn't really creating the CDKN2A deletions, but it's certainly enhancing its effects. Apparently, tumors that are able to acquire this event are better able to deal with the toxic effects of radiation. So in addition to the hypermutation and the microdeletions, this is a third category, if you will, a third type of molecular abnormality that we can identify as being associated with tumor progression in IDH mutant glioma. And if you summarize this across our current GLASS database, I won't go into too many details, but I think this is well over 100 cases. I don't know the exact number, actually. But if you summarize this, we find that in 60% of tumors, 60% of IDH mutant gliomas, we can identify one of these categories of mutational events that are acquired during tumor progression, and often multiple in the same tumor. And this is actually not unimportant, especially if we evaluate survival. So if you look at overall survival from time of diagnosis and compare tumors that end up acquiring at least one of these types of events versus those that remain genomically relatively stable between diagnosis and recurrence, we see that cases that end up acquiring a genomic event do much worse and have much worse overall survival. So it really suggests that this model is in place. Again, we are starting from a cell of origin that gives rise to a multi-clonal or multiple subclones within the tumor by the time the tumor gets diagnosed. Through treatment, we chop away a number of these subclones, but there are some cells that were already present at time of diagnosis or that are being created that acquire events after time of diagnosis. And those are the cells that are then able to repair DNA damage projected by these treatments. And those are then the cells that we find at the time of recurrence. And we know it has to be those cells because of these molecular signatures that we are identifying. And the cancer clones that are large enough to be detected through sequencing are the ones that, that we see at recurrence. So it's a clonal selection model driven in part by treatment. Okay, so far all published data. Now we're getting to the fun part. So we've had a really fruitful collaboration over the past couple of years with 4 other labs in totality. We collaborated with Mario Suva at MGH, Itai Taraj at the Weizmann, Antonio Iavarone and Anna Lazzarella, who are both now at the University of Miami. In collaboration with them, we acquired a large number of tumors, both IDH mutant and IDH wild type. I'm only going to talk about the IDH mutants today. We acquired multiple specimens from those tumors over time. We collected clinical annotation, but most importantly, we generated DNA sequencing, either whole exome or whole genome sequencing, and single nucleus RNA sequencing of at least 2 time points. And we did this for 35 patients diagnosed with an IDH mutant glioma, in total 75 samples. So for some patients, we had more than 2 samples. For a subset of the cases, we actually did multiome, which means we did single nucleus RNA-seq, but also simultaneous single nucleus ATAC-seq. And as I said, we have DNA sequencing for pretty much the entire cohort. So this collaboration involved a very large number of both PIs and analysts, where Kevin Johnson really was the leader of the analysis that I'm going to talk about. And Kevin is a research scientist in my lab. With this large data set derived from IDH mutant gliomas in hand, the first step that we did was separating cells that are malignant or cells that are non-malignant. So we are separating the tumor microenvironment from malignant cells. I won't go into the details of this process. I'm happy to talk to you about that if that's something that interests you. Just suffice to say that we had a very strict quality control process, and we actually tossed out about 40% of the cells that we started with to make sure that our subsequent analysis was as accurate as we could make it. So I'm very confident to say that what we label here as a malignant cell is truly a malignant single cell. So we then did UMAP analysis to separate different clusters or different groups of different cell types separately for the codels and the non-codels, the oligos and the astros. As you can see, even though we did a very stringent quality control process, we still had a very large number of cells remaining. And as you would expect, we find that the majority of single cells are single nuclei, but I'm going to use single cells from now on as a term. The majority of the cells are actually malignant cells, but we find oligodendrocytes in the microenvironment, we find myeloid cells in the microenvironment, all the cell types that you would expect to see there. So one of the first questions we asked is, amongst the malignant cells, now with a much larger number of cells in hand relative to when Itai and Mario did their 2017 analyses, can we go more into depth? Can we identify cell states, and even rare cell states, just because we've got this massive data set? So to do that, we used what I think is a fairly complicated computational analysis that was invented by Itai and his group, which essentially is running NMF multiple times. It actually took about 4 weeks to compute one single iteration of this plot. And at the end of that, we can say that all the malignant cells can be binned, or more or less all the malignant cells can be binned into one of these categories of cell states. And these cell states were labeled OPC-like, AC-like, mesenchymal-like, and NPC-like based on the expression of marker genes in their respective gene signature. So 4 cell states, and then finally we also have a orthogonal layer of cells being actively cycling, yes or no, again based on the expression of particular marker genes. Now you might say, hold on, I've actually heard of these terms before, and you would be, of course, dead right, because Mario and Itai published a very influential paper in 2019 where they reported the exact same labels for 4 dominant cell states detected in IDH wild-type tumors. And that's, of course, no coincidence. Even though we had a very large number of cells, our, what I think of as very robust analysis, really identified 4 cell states that are highly similar to the ones that were derived a couple of years ago for IDH wild-type tumors. So even though genomically IDH mutants show a large variety of abnormalities between the 2 types of IDH mutant tumors, and IDH wild-type tumors are different altogether. When we look at the cell states, when we look at this, the gene expression programs that drive these tumors, they're actually highly similar, which I think is fascinating. Now, in parallel to identifying gene expression signatures and gene expression programs, we can still project each of those cell states on the inverted y-axis, on the inverted y diagram. So that's what we did that here, and this is to visualize the level of stemness of each of these 4 gene expression, each of the 4 cell states. So we can then, for example, conclude that the NPC-like and the OPC-like cell states, or at least the NPC-like cell state is higher up the inverted Y. In other words, the NPC-like cell state is more of a stem-like cell state compared to, for example, the AC-like cell state. There's also a new category introduced here, and that's a cell state that we labeled undifferentiated. And this is basically all the cells that did not neatly fit into any of the 4 bins of NPC-like, OPC-like, MES-like, and AC-like cells. So we really have 5 categories of cells, with the undifferentiated cell being the ones that do not fit. But as you may be able to appreciate, for both within oligos as well as within astros, this is a fairly stem-like cell state. As previously, as we saw a couple of years ago in the original publications, we see that the highest level of cycling activity is, of course, found in the more stem-like cells. No big surprise there. And as you could have inferred from the previous slide, the 3 cell states that have the highest level of cycling activity were the undifferentiated cells, the NPC-like cells, and the OPC-like cells. And this is true for both oligos and for the astros. And actually, we're starting to see a bit of a dichotomy in cell states, and I'm going to argue that I'm going to substantiate that further. But there are 3 cell states that are clearly more reflective of early cells, progenitor cells, and stem-like cells. And there are 2 cell states that are really more differentiated-like, and that's mesenchymal-like and AC-like. Because we did multiome analysis, so we did simultaneous ATAC-seq as well as RNA-seq on about 2/3 of our cohort, we could then also evaluate chromatin accessibility across the different cell states that we had characterized. And by doing that, we can further support that there's somewhat of a dichotomy going on in between these different cell states. Again, showing that the undifferentiated cells, the NPC-like cells, and the OPC-like cells are more stem-like and show more similarity to one another, as you can maybe see at the top here. Whereas the AC-like and the MES-like cells are grouped more or less separate from the other 3 states because they are more differentiated. We can, of course, mine the chromatin accessibility data to identify key regulators of an individual cell state. So we And we do that by contrasting chromatin peaks across many cells. And when we did that, we find we predict master regulators for these cell states, for example, ASCL1 or TCF12, or FOS and Jun for the MES-like state. Now, if you've been following the literature on key regulators, these are not newly identified master proteins. In fact, these are in line with what have been reported by others. And it again comes back to the dichotomy that I've described before, where we see sort of a gliogenic switch occurring with the master regulators of each of the cell states being more or less aligned along this glial differentiation trajectory, with cells being pushed more towards an oligodendrocyte lineage, more towards an astrocyte lineage, or being contained in a more progenitor-like state. So our data is nicely converging with what is actually known in literature here. We evaluated how different grades compare to the presence of the different cell states. So, starting with the IDH mutant oligos, we see that, and for oligos, of course, there's only grade 2 and grade 3. So we see that a grade 3 oligo has a much lower fraction of cells labeled as AC-like, so fewer cells that are differentiated, whereas we see a significant increase in the number of undifferentiated cells, and a borderline significant increase in the number of cycling cells. When we look at astrocytomas, we see a similar trend. For astrocytomas, we recognize grade 2, 3, and 4. So clearly, a grade 4 astrocytoma that is still IDH mutant has a much lower fraction of AC-like differentiated cells, an increase in the number of undifferentiated cells, and also an increase in both mesenchymal cells and the number of cycling cells. Now, when we do the longitudinal comparison, so the previous slide was just binning all our data by grade. Now, in this figure, we're actually matching samples by time point. So when we evaluate longitudinal shifts, longitudinal potentially transitions in cell state distributions, we're seeing the same trend towards dedifferentiation, an increase in the mesenchymal-like signal activity, and a significant increase in the number of cycling cells. And this is independently true for astrocytomas and oligodendrogliomas. And because of that shift towards dedifferentiation, when we project our tumors, we basically project the median cell state across an entire tumor. And we do that in a pairwise fashion for longitudinally linked samples. Of course, we see the expected shift upwards on the inverted y-axis, so tumors become increasingly more stem-like, less differentiated as they progress. Now, if there's one thing that I would like you to take away from what I'm talking about today, it's actually this slide, because this I think is actually our key result from the entire analysis, and that is that I talked earlier about how we see about 60% of tumors are acquiring a genetic abnormality that falls in one of a number of classes, namely hypermutation, microdeletion, or cell cycle alteration. When we now group our single cell cohort by tumors that have acquired such a genetic alteration, and another group of tumors that has remained stable, that did not acquire an alteration in any of these categories, we very clearly find that the shift towards dedifferentiation is driven by those tumors that have acquired a potentially therapy-induced genetic alteration. And remember, I say therapy-induced because hypermutation is linked to temozolomide, and both CDKN2A loss as well as microdeletions and large increases in aneuploidy are linked to radiation. So we see that this is the group of samples where we see a marked shift and significant shift in AC-like cells, undifferentiated cells, and cycling cells, and we do not see that same shift occurring in samples that have stable genomes. The exception here is in the number of mesenchymal-like cells. So mesenchymal-like signatures, the number of cells classified as mesenchymal, do increase regardless of whether there's a concomitant genetic alteration acquired, yes or no. So we then turn to GLASS to try and validate this result. So in GLASS, pretty much all tumors have whole exome or whole genome sequencing data, and a subset have matching RNA-seq data. So we took that RNA-seq data, we used computational methods like CyberSort to deconvolute the bulk RNA-seq profile into the single cell-derived counterparts. And when we do that, we see the same shifts. So this is an entirely separate cohort. We see the same shifts in a decrease in AC-like cells. Actually, not a significant shift in mesenchymal-like cells, and I'll get back to that, but the same increase in undifferentiated signature with time point. Now, why do we not see a mesenchymal-like shift? That's because when you take a bulk RNA-seq profile, the mesenchymal signature is too subtle to be picked up. So we are not able to retrieve it by looking at bulk RNA-seq. We really need single cell or single nucleus data sets to identify the mesenchymal signature. But again, when we group our GLASS cohort into tumors with or without therapy-induced genetic alterations, we very clearly see that the transitions that we are finding are driven by the group of samples that have genetic alterations acquired. So there's a strong convergence here. Now, as I've mentioned several times, both in our data set as well as a separately compiled data set of public RNA-seq data, we see a significant increase in the cell state, in the fraction of cells marked as mesenchymal-like. And when we cross-reference our cell states, our malignant cell states, with the fractions of cells in the tumor microenvironment, so here we see the malignant cell states on the y-axis and a number of different tumor microenvironment cells on the x-axis, we see a very significant signal between the number of mesenchymal-like cells and the tumor-associated macrophage bone marrow-derived myeloid-like cells. That's a long acronym. But these are really myeloid-derived macrophages in the tumor microenvironment, and we see a very strong correlation between those two. So that shift towards tumors increasing the number of cells that are mesenchymal, the number of malignant cells that have a mesenchymal phenotype, converges with the presence of myeloid cells in the microenvironment. And of course, then it's a chicken and egg story. I mean, are the myeloid cells driving a mesenchymal feature, or is it the other way around? And of course, we cannot really say one way or the other. But there is definitely a extrinsic factor associated with the mesenchymal signature. So final few slides. So I'm going to talk about a few specific genomic abnormalities and how they relate to different cell states. So focusing on 3 abnormalities that we find frequently in IDH mutant tumors, namely homozygous loss of CDKN2A, focal amplification of PDGFRA, which can be and often is extrachromosomal, and hypermutation. So we find that in comparing tumors with and without either of these alterations, we see a significant change in the number of undifferentiated cells, AC-like cells, and cycling cells. Now, why am I highlighting this? That actually requires slightly more introduction. So coming back to temozolomide and hypermutation, so temozolomide is a dealkylating agent, and as a result of the DNA damage repair process of dealkylation, there can be a C to T change occurring in the genome, and that's what actually is the source for hypermutation. Now, it's very challenging to call specific mutations in single-cell data, and that's because single-cell gene expression is essentially 3' end tagging. In other words, you're only marking a very small amount of actual coding sequence of each gene. But because the shifts, the mutations that are driven by temozolomide are so specific because they contain this specific signature of C to T. Instead of looking at individual mutations, we can enumerate the number of C to T changes in the territory that was sequenced for each single cell. So we did this and then compared the relative fraction of C to T mutations in non-hypermutated and in hypermutated tumors. And you can see that in tumors that have hypermutated, and these are each single cells, we can clearly identify a large number of cells that have a relatively high C to T burden. And we could repeat this in the simultaneous single nucleus ATAC-seq data to validate our finding, and this very much validates. So 99% of the predictions based on RNA-seq are validated in the ATAC-seq. So we can pick a really nice threshold somewhere in the middle and say, and then label cells as being hypermutated cells or non-hypermutated cells. And then we can do gene expression comparisons within a tumor. So we did this, and then looking at comparing hypermutated cells versus non-hypermutated cells within a tumor, we see the expected transitions, or the expected differences, I should say, with cells containing a high C to T burden being less AC-like, more undifferentiated, and more cycling. And consequently also significantly different when projected on the inverted y-axis. So this, I think, is potentially an important result because it argues that we're precisely following this model, where it is the cells that have the ability to repair DNA damage that are really driving the progression of the tumor, and driving the progression of the tumor in part because they're able to de-differentiate the cells and become more stem cell-like, and therefore giving rise to more proliferation. Now, calling homozygous deletions in single-cell data is extremely challenging, but calling focal amplifications is not impossible, and that's because an amplification will give rise to higher amounts of sequence. So we used a method called copyscat to call focal amplifications using our single nucleus ATAC data, and it's a relatively straightforward method that basically looks for a change in the amount of coverage across the genome, and then you can label that as a focal amplification. And of course, we had a starting point. We knew, based on our whole genome sequencing data, exactly which tumors contained a PDGFRA amplification and also what the territory of that PDGFRA amplification was. So we could perform this analysis in a somewhat supervised way. And doing that, we can use the ATAC-seq data to classify a cell as containing a PDGFRA amplification, yay or nay, and then validating that result by evaluating PDGFRA gene expression from the single nucleus RNA-seq data. So you can tell that in some tumors that works quite well. Here's the gene expression of cells with or without predicted PDGFRA amplification, and you can see that in this tumor, those cells that have predicted amplification have a much higher level of expression, whereas in other tumors it may not be working as well. So it's certainly not a perfect analysis in that sense. But following this this dichotomy, and here are this is the relative. It's hard to read from my angle. This is the relative proportion of cells that are marked as being cycling, yes or no, amongst the cells with and without PDGFRA amplification. And as may be expected, in at least 4 of the 5 tumors, we see a higher fraction of cycling cells in cells that contain PDGFRA versus those cells that do not. And similarly, we see a relative decrease in AC-like and a non-significant, but it's only 5 tumors, increase in undifferentiated cells. So again, following potentially that model where the cells that contain the PDGFRA amplification are potentially the ones that are actually driving the progression of that tumor to become larger and more, giving rise to more symptoms. Okay, so on that I would like to end. So to summarize, I hope to have shown you that the cell states that we have defined for IDH mutant glioma are reminiscent of IDH wild-type cell states. That's actually only possible because we combined newly diagnosed and recurrent IDH gliomas in our data set, because we really see a much higher fraction of mesenchymal-like cells in recurrent gliomas. I hope to have shown you that IDH mutant glioma progression is really primarily driven by acquired genetic alterations. You saw the very significant difference in overall survival in the Kaplan-Meier curve. These acquired genetic alterations result in cells becoming, in driving dedifferentiation expression programs. Cells become more stem cell-like. And then finally, a secondary axis that appears to be operating in parallel is a shift towards a mesenchymal-like state, which converges with an influx of myeloid cells. And maybe a final comment also based on what we were talking about just before. It would be extremely interesting to perform a similar analysis, but now on tumors treated with vorasidenib, the new IDH mutant inhibitor. There was a small paper published on this just a few weeks ago by Mario and Itai that seems to suggest that in a tumor that responded to vorasidenib, the tumor became more differentiated. So that gives rise to all kinds of hypotheses that I would love to explore further at some point in the future. But for now, let me thank the lab, in particular Kevin Johnson, who drove this work, and of course our collaborators that I've talked about before, our funding agencies, and happy to take a few questions. Thank you.
Thank you. That's very informative. We work on GBM, so this is like kind of a prior state, and so I was interested for the treatment that they undergo these patients, and if there's some information on your studies that show that the mutations that they acquire, either hypermutations or microdeletions, they lead to reversion from IDH mutation to IDH, so wild type, and therefore to GBM. And if you think that's sort of the consequence of the treatment.
So I'm going to repeat the question because I think it's recorded, and otherwise you won't show the recording. So the question is whether we find that the acquired genetic alterations can result in the tumor becoming IDH wild type. And the answer is that we never see that. We never see the actual IDH mutation disappearing at recurrence. It can be a little tricky to call the mutation from sequencing data, but when you look into in detail, basically if you look at the raw sequence, you pretty much always will find the IDH mutation. That's not said that the effects of the IDH mutation couldn't be disappearing, right? We know that the primary effect of the IDH mutation is the 2HD to 2kg, if I have it correct. So it might be that at recurrence, that oncometabolite may be much less present. We don't really have the data to measure that. That would be certainly an interesting question. If you look at the cell states, however, and maybe also to some degree at the genomic landscape, there's certainly a truth in that a recurrent IDH mutant glioma more resembles an IDH wild-type tumor. Potentially one big difference is the level of aneuploidy, where an IDH wild-type tumor already at diagnosis is usually highly aneuploid, and we see a much lower level of aneuploidy in general in IDH mutants, including at recurrence. So.
Very nice. So the mesenchymal shift, so you obviously see it overall, but I noticed that it was quite heterogeneous, so in a lot of patients it didn't actually change. So do you know does that correlate with the influx of myeloid cells, for example? Do you know why there is such degree of variability? And the second question was, what do you think that does? So that the mesenchymal shift, at recurrence, does it actually have a biological impact on recurrence? Do you think these cells are driving anything specific?
Okay, so question one is what drives the increase or maybe the non-increase in some tumors of mesenchymal-like cells? And second, whether it has a biological implication. So, well, we clearly see a very strong correlation between the number of mesenchymal-like cells and the number of myeloid cells. So there's some interaction there, and as I said, I mean, it's chicken and egg. We don't know if which precedes the other, essentially. To some degree, I think of myeloid cells as going into the tumor and cleaning up debris. And it could be as simple as a sort of I'm not saying that it's simple, but maybe it's almost like a wound healing response. Following the surgery, myeloid cells go in and try to clean up the mess or the injury left behind by the surgery. And maybe there's even neurosurgical factors we could try to evaluate in association to whether the tumor is likely to acquire these myeloid cells and subsequently mesenchymal cells. Whether it has a biological effect, I can only speculate, but because we see in many tumors that we see an increase as the tumor progresses, and I didn't show that, we also know that at T2, when we split the cohort by the relative fraction of mesenchymal cells, tumors that have higher levels of mesenchymal cells have worse outcomes. So it does suggest that it's either a biomarker or an actual driver of a lack of response to further treatment.
Go with Lucy first. I think she was first.
So I'm interested in the connection between the hypermutation and the increase in the cells in this undifferentiated state. So do you know what could be driving it? From what I understand, you can't look at specific mutations with this approach, right? But could you look in the RNA-seq at different pathways which might be altered?
Okay, so why do hypermutated cells become more stem-like or less differentiated? It's a great question. We've only evaluated cell state level differences. We could go into the data set and ask the exact question you're asking, like what are pathway level differences, but we haven't done that yet, so I can't really speak to that. But just speculating, I mean, whether it's pre-existing or it's acquired, I don't know, but these cells have more ability to repair the damage done by temozolomide. And maybe as a consequence of the ability to repair that damage, they are also able to activate a more proliferative phenotype. There's probably a convergence there.
So I was just trying to figure out whether just trying to understand, maybe I missed it, is that the initial tumors that are primary tumors that you have, so you know the genomic burden of those tumors, and then they progress over time. This is probably post-therapy. Have you looked at then the tumor evolution? Do these primary tumors have a predisposition to certain states? And can that be linked to certain genomic alterations? And then, because you already have the GBMs, can you have like a more global thinking of, because it seems like the IDH are now seeming more resembling the wild types. And do you think it's a genomic change that could be predispositive?
Yeah, so I think what you're asking is, can we predict which tumors are going to be undergoing these de-differentiation, becoming more mesenchymal-like or more stem cell-like? So I think to me, that's now the million-dollar question. So what I showed here was our data sets that we've accumulated with single nucleus RNA-seq or single cell RNA-seq data. But I also briefly mentioned the glass cohort, which is in the end a much larger cohort. By now, I think we have 650 tumors in there, of which probably 250 to 300 are from IDH wild-type gliomas. And I'm really excited about that data set, which we haven't really reported in literature yet, because a large number of those tumors we've whole genome sequenced. And I'm hoping that we can use mutational signatures that we detect at the time of diagnosis to predict which tumors are more likely to become hypermutated or that are more likely to acquire microdeletions or CDKN2A loss. But we haven't been able to do that analysis just yet. But the thinking there is that a mutational signature is derived from a certain DNA damage process, and that if a DNA damage process is active in a tumor cell before diagnosis, maybe that's a predictor for that DNA damage process to actually be driving future evolution. So.
Yeah, like even the ones that you showed where you saw the non-responders to the responders, and then
right.
You have the IDH mutant there. Is there anything genomically in the evolutionary tree that kind of identifies a certain clone that is from a particular genome?
Yeah, so far we don't see that yet, although the number of cases is relatively small. Amongst IDH wild-type tumors, we do know that it's only the tumors that have MGMT methylation that become hypermutated. So there's a clear predictor there. Yeah. Which makes sense because those are the only tumors that respond anyway, right? And you can't really form a hypermutation unless the tumor actually responds to a certain degree. Right? So in that sense, these genetic alterations are also markers of a tumor that actually had a response to treatment.
Yes, please.
Thank you. That's a really interesting talk. I was wondering about the potential germline component, because you mentioned the Bob Jenkins paper, and then they and others showed that risk regions identified by genome-wide association studies often show very extreme preference to either IDH mutant or non-mutant tumours. I didn't know if this data set and others as part of class could help explore that, and particularly like the FT24 region.
Yeah, so what's the contribution of the germline? So I think it's a very interesting question. We were just talking about, before the seminar, we were talking about that and how it's potentially not enough, has not been sufficiently explored yet. The variants that have been reported in case control studies, their effects are significant, but they're really weak, right? You're talking about a variant that gives an individual twice the chance of GBM relative to somebody that doesn't have the variant. So you take an extremely small chance, and now you've got twice the extremely small chance, right? So those odds ratios are still, I think, very low, beyond sort of the genetic syndromes like Lynch syndrome and NF1, etc. Having said that, I do believe that there are probably very powerful variants that we haven't detected yet because they're so rare across the population. So I do think that there's still a lot to learn from germline analysis, because I do think very powerful variants exist. We clearly see clusters of tumors in families. And every clinician in this room and elsewhere knows at least one or more families that have at least two members with a glioma or glioblastoma. So there's more familial component than we often consider. So, yeah.
Perhaps a final question.
Hi, very nice talk. So I have a question about undifferentiated cells. So if I understood correctly, they are non-cycling, right? Because you divided the cycling cells.
They actually have a fairly high fraction of cells that are cycling. So they're undifferentiated and cycling.
Because my question is more about the differentiation. So here you're talking about proportional cells. So how much evidence you have about the differentiation process, whereas, for example, cycling cells starting differentiating again and doing undifferentiated populations.
Well, that's a fair comment. So how do we know that we're actually seeing de-differentiation? And I think we really can't, because we're taking a snapshot at one single time. So in that sense, we should probably be cautious in using the term de-differentiation. What we can say is that we see a proportional difference in the number of differentiated cells. And how the tumor gets there, whether that's de-differentiation or whether undifferentiated cells just cycle more quickly, it's something that will be interesting to find out at some point.
Great. Well, you've made me nervous about giving my patients temozolomide, so thank you very much. If we could all come together to thank